So in this video, we're going to talk about enzymes. And enzymes are biological catalysts. And they are hugely important to pretty much all biological functions are catalyzed by an enzyme. But let's get to basics here. What is a catalyst? So a catalyst is something that speeds up a reaction. And I'm going to abbreviate reaction RXN, but is not used up in a reaction or in the reaction. So a catalyst is not just a biological thing. There can be all kinds of catalysts in all kinds of processes. In biology, an enzyme, a biological catalyst, is either, most commonly, it's a protein, but occasionally it can be an RNA molecule. So RNA is another type of molecule that has that can have the ability to catalyze reactions, and we're going to talk a lot more about that later in the course. So let me kind of show you how an enzyme works in a very cartoony way. So I'm going to draw some molecules here as circles and triangles. Here's another one. Okay, and they're all bouncing around in a solution together. And those we're going to call, those are our reactants. Okay, and then over here I'm going to draw our product, or our products. So this is a chemical reaction. It starts with these guys, and it ends with a molecule that looks like this. So the triangle has been joined to the circle in a specific way. Now, this reaction can proceed without the enzyme. It requires the reactants, and remember, the reactants have kinetic energy. They're bouncing around. Kinetic energy just means that they have motion. They're bouncing around. They're colliding into each other. And every once in a while, when the collision is of the right amount of energy and the two substrates or the two reactants are oriented just right to each other, boom, this bond can spontaneously form and produce this product. But the probability of that happening is fairly low, and so it takes a long time for these reactants to combine in this way to form the product. So the way that biology solves that problem of rate, the fact that reactions take place quite slowly, is through the production of something called an enzyme. And like I said, an enzyme is a protein. I'm going to draw it like a blob here. And it's a blob with a cunning little pocket in it that we call the binding site, that just so happens to fit our two reactants just perfectly. So here's our enzyme. And what happens is, all along this binding site are residues or chemical, surfaces that bind really, really nicely with these two reactants. And so what happens is this triangle binds quite easily in this pocket here. Oh, I didn't draw this quite right. And the circle binds really easily in this pocket here. And because the enzyme has a specific orientation to those two substrates, the chance of them forming that bond becomes greatly, greatly increased. So basically, it creates a two-step process where the reactants have to bind to the enzyme in that binding site that catalyzes the production of the product. You may have noticed that I'm kind of using the term substrate interchangeably with reactant. So you call a reactant a substrate when you're talking about an enzyme catalyzed reactions. So enzymes work on substrates. And this is called the enzyme substrate complex when the enzyme is bound together. So, just as a quick review, enzymes do speed up reactions. And the way that they speed them up is that they increase the probability that a reaction will occur. Okay. The other thing that they do is that they decrease what's called the energy of activation. That's a lot. That's a big mouthful, and I'm gonna abbreviate that to E sub A. They decrease the energy of activation of a reaction. And what that means, the energy of activation is simply, you can think about it as a little energetic barrier or an energetic hump that the substrates have to overcome in order to then proceed with the reaction. And I can draw that for you in a minute. But what enzymes do not do, and this is a common source of confusion for students, enzymes do not add energy to a reaction. And this can be a little bit of a subtle point. So if you think about a reaction where you're starting with a substrate and you're building a molecule that has lots of energy in its bonds, the enzyme cannot provide that energy. That energy has to come from one of the substrates. It has to come from another molecule like ATP. In the case of photosynthesis, it has to come from sunlight. It has to come from somewhere, because the enzyme, remember, the enzyme is not used up in the reaction. The enzyme is not altered. It starts in a certain state, it catalyzes the reaction, and it goes right back to that same state. It hasn't been changed at all. It hasn't added energy. It hasn't taken energy away. It just sort of is. And so, but what enzymes do is they decrease the energetic hump that these molecules need to go through in order to get to these products. A way that you can draw that is, I can draw you a couple of graphs here. And this will be the reaction progress. So this axis is almost like time. And I'm just going to call this energy for now. Energy, so these x. So basically I'm going to draw the energy state of a reaction as it moves through time. And so reactions can start at a low energy state, and then the products can be at a higher energy state. So these are the reactants here, and here's the products. And this is the energy that needed to be added in order to get that reaction to go forward. The difference between the reactants and the products is what you need to add. But notice that you actually have to add just a little bit more. And that's the energy of activation. So it's a little bit of extra energy that you have to kind of push to get the reaction to go. So similarly, we can have a reaction in which the reactants are a higher energy state, and then the products are down here. So I'll just write reactants, products. And in this case, this is a reaction that's releasing energy. But notice, again, there's a little energetic hump here that has to be moved through in order for the reaction to go. So what an enzyme doesn't change the overall energy state of the reactants or the products. What an enzyme does is it changes the height of that hump. So for example, the height might be like this without an enzyme, and then it goes down to this with an enzyme. Similarly, it might be without an enzyme, that energy hump might be bigger, like that, but with the enzyme, it brings that down, or maybe even it brings it down even a little farther. Maybe it makes it like this.